So I'm going to be speaking at this meeting on novel combinations in relapsed disease and trying to achieve minimal residual disease, MRD, negativity. We're very excited in CLL to have so many great new tools. These new drugs have been very effective as single agents, but now we have the opportunity to put them together to try to achieve deeper and more durable responses, even for patients with relapsed CLL. So I'll be starting by talking about treatment of relapse-refractory CLL, and in particular using novel combinations. And one of the things that I'll be discussing is dual blockade of the B-cell receptor pathway. In other words, using a drug like ibrutinib in combination with a drug like umbralisib, a PI3 kinase inhibitor, together. And we think that this could be a very well-tolerated and effective strategy. We also are going to be discussing data combining ibrutinib with venetoclax, so probably two of our most potent drugs targeting the BTK protein as well as BCL2, and now using these together. And we have several great examples from recent trials that have been presented or published. One of the most challenging problems for us in CLL right now is the development of Richter syndrome, an aggressive transformation of CLL. So I'll also be presenting some of our recent data combining venetoclax with chemotherapy, with venetoclax and our EPOCH for Richter's patients specifically, as well as some other promising studies being conducted around the world using checkpoint blockade antibodies such as pembrolizumab or nivolumab, given in combination with BTK inhibitors. And then finally in this talk, I'm going to explore novel combinations of drugs targeting other apoptotic proteins. So BCL2 is just one of many proteins in that family. So we'll discuss the future possibilities of targeting proteins such as MCL1 and BCL-XL in combination with BCL2, which we think might also be a very effective strategy for CLL patients.